this video is going to pick up with question 19 in the practice air test for biology. Looking at question 19, a scientist is trying to find a relationship among species that are very similar physically, and one way you can do that is by looking at amino acid sequences. Amino acid sequences, that means the actual chain of amino acids that make up proteins. Why is this useful? Why can we do this? So which one of these answers gives the best explanation for that? And the best answer for this is A. I'm sorry, not A. The best answer for this is B. Amino acid sequences, or sequence differences, reflect the accumulated differences in the DNA of the species. So amino acids, remember, are the product of RNA, and RNA is the product of DNA. So because DNA mutations, DNA changes over time can lead to amino acid changes over time, looking at the amino acids is almost like looking at the DNA. So looking at those two things, either one of them, can give you some evidence to show you how closely related species are. The more similar the amino acid sequence, the more similar the DNA sequence, the more closely related the species probably are. The more differences they've developed over the years, the more distantly they're probably related. Again, that applies to DNA and amino acids. So the best answer here is B. Next up, which two of the following form the base pairs in DNA? We're talking about DNA specifically. And remember, this goes back to Chargaff's rules. He said that A pairs with T and C pairs with G. A and G no, AC no, GT no, CT no. A and B here are correct. A and T pair, G and C pair. Looking at a cladogram here, and in this cladogram the question is which conclusion is supported. We're looking for, in this case, remember, a cladogram is showing you relationships, and this is modern. This is where we have all of our names here. These are the different species. This is now, and as you go backwards to the other side, this is back in time. So what we're looking for is when these species split from each other. So we see a split happen here, where Cupulata split away, and then we see a split happen a little bit later, where Valerioi split away, and then we see the last split happen between Rosea and Cylindrica, and so this one is the most recent split. So this split was a little older, and then the split that happened back here to split this one away from the rest is even older. So looking at our four answers, the most closely related, the Valerioi, I'm not good at saying that one, and Rosea, those are not closely related. They're not the closest. This is the one. Rosea and Cylindrica are the most closely related. B is our best answer there. These two being closest related, no. Again, they're close together on the chart. That does not mean they're closely related. We have to go backwards until these guys meet, and where they meet is way back here at the beginning, so they are not closely related. And then same thing with these ones. They don't meet until they come way back here. So, closely related, Rosea and Cylindrica. Why? Because they've split the most recently from each other. So that's letter B. Next up, this is about population growth. Serengeti buffalo, they had a virus, and so a bunch of them died, and then what was remaining in the population, over time, they built their way back up again. The population grew back when they recovered from this virus, and it hit kind of a limit, and then after it hit that limit, it started to sort of bounce around in this general area. So the question is, which of these numbers, of these quantities for the population, seems to represent what we would call the carrying capacity for these buffalo after the virus was kicked, once the virus went away. So 35,000 is way too far down there. 55,000, we're getting close. 55,000 hits right here. 60,000 is right here. It's kind of right in the middle of these spikes, up and down, go around that. And then 65,000 is the peak. You might think that the peak, the highest one, is the carrying capacity, but we don't call it that. We call this big spike that usually happens at first an overshoot, because then there's a correction right after and it drops back down again. So when this thing starts to get kind of wavy, the carrying capacity is right in the middle of that wave. So 60,000 was actually a better answer for this one. So that should be C, 60,000. Next, we're looking at two types of mice that live in a valley together. One type has webbed toes, the other one does not have webbed toes. There's a valley built, and so the part of the valley floods, it turns into a wetland. As the years go by, if the biologists looked at the mice later on, they found that they were all webbed toes. So the two types have now changed. So after some generations go by, all the mice are web-toed. There are no non-web-toed mice left in this area. So what's the best explanation for this? This sounds like a case of natural selection. Natural selection meaning the best traits are the ones that linger, and the traits that don't do as well are the ones that disappear. In this case, we're looking at answer C. The web-toed mice, they're the ones that survive better, and that means they reproduced and made more mice than the ones without web toes. And so eventually, the ones without web toes diminished until eventually they disappeared completely. The other ones don't really make as much sense, but again, natural selection works like this. The ones with the better traits survive better, and they reproduce more offspring. And so C is the right answer. Next. Alright, this is an electrophoresis. What we're doing here is we take these things called restriction enzymes, we chop up the DNA with them, they're like little scissors, they cut the DNA in certain spots, and different DNA strands get cuts in different places. So depending on the source of the DNA, we might see different groups, and so these represent, typically we start at the top here, and the DNA actually slides through this stuff. It's like a gel, like Jello, and the smaller the pieces, the further they go. So what we see here is like a little small piece of DNA, and then a little bit bigger, a little bit bigger, a little bit bigger, and then like one that's really big up at the top that didn't go anywhere, so it didn't slide very far. So depending on the sizes of each piece of DNA, we can see how far they run down through this gel. So what we're looking for is, this is rat A, and we want to know the potential relationships of rat B, rat C, and rat D back to rat A. So how does rat A compare to these three? So what we're looking for is, are they unrelated, are they identical siblings like a twin, or are they a different sibling or a parent? So they're related but they're not identical. So remember, identical twins have the exact same DNA, so we should expect to see an identical banding pattern. So rat C and A, if we look at all of the bands, they all match. So rat C must be identical with the DNA to rat A. So an identical sibling is rat C. If we're related, like a parent or a sibling, typically, because you get half of your DNA from mom and half of your DNA from dad, typically you would see around half of the bands that would match. You would see somewhere around half matching. So we see this one matches with D. We see one of these two matches with D. That one's different, but this one could be matching here. It looks like it matches. This one matches. We see again that one doesn't. This one doesn't. So that's about half of them matching and half not. So rat D makes the most sense to call that one a sibling or a parent, someone who's related. And rat B seems to not really match at all, even where we have lines that seem to match. This is a pair of bands, this is just one. These guys are not here. These don't line up the same. This seems to be an unrelated rat. So there's our three answers for that one. Next up. In this population, we're looking at incomplete dominance. Incomplete dominance, remember, is when we have two extreme phenotypes. In this case, we have light brown and dark brown. And sometimes if we have a heterozygote, that means they get one of each copy of those alleles. The heterozygous can end up somewhere in between. So in this case, that would be a medium brown color. These are stone crabs. An introduced predator favors the medium brown crabs. Maybe they stand out the most against the rocky background, but the medium ones seem to be the most grabbed by predators. So the question here is, how would this selection pressure, how's that going to affect the relative frequencies? That means like how often we see the big B, the dominant, or it's not really dominant, but the big B allele, and how often we see the little b allele. So as generations go by, we're actually going to see not a big difference because the medium brown crabs have one of each. So every time a medium brown crab is eaten, we're losing a big B and a little b at the same pace. So what's going to actually happen is kind of a weird question, actually. You would think they'd do something that would change, but in this case, you wouldn't actually expect a change in either one. Because, again, the heterozygous individual, the big B, little b one, is, if it dies off, you're losing a big B and a little b each time, so you should see, relatively, the amounts are going to stay the same. So no change for each one. Alright, this will be our last one before I cut this video off, and then take a break and do the next one. What we're looking at is a cladogram that we need to fill in. We see a chart listing amino acid differences. Remember, the relationships between organisms, the closer they're related, the fewer differences we should see between the amino acids or the DNA. So in this case, we're looking at how many differences are there in some 113 amino acid chain. It's 113 amino acids long, and we're looking for the smallest number of differences to be the closest related organisms. So there's a couple ways we can go about figuring this out, but one of them is to look at individual D here. We see D has a close relative, has one close relative. D is more distantly related to these other two, and even more distantly related to the last one here on our list. So if we find the closest relative to D, we should find the one with the fewest differences from D. So if we go down the column here, D is 11 away from A, 9 away from B, 8 away from C, but if we look the other way, D and E are only 3 apart. So it seems like D and E should be neighbors here. They're the ones that are closely related. We should also expect to see 2 that split very recently, so they should be very similar. So I can figure out these ones by looking for the smallest differences on here, and we see 1 difference between B and C. So B and C should go right here. And that could be BC or it could be CB, it doesn't matter. But they go together because they have the fewest differences, so they're on the closest, most recent split. A is the only one left. A is 11, 10, 11, 14 away from everybody else. Those are big numbers compared to the rest of this chart. So A splits really early on. So A is the least related to the rest. So all these are in the right spots. And we will stop here for this video.